Good evening and welcome to Armor of God Apologetics. I'm Dr. Wayne Harship. As a quick recap, we have moved on from the cosmological argument for God's existence, which holds that the origin of the universe indicates a creator in an analogous fashion to how the presence of a building by logic implies a builder, or how the presence of a beautiful painting implies a painter. Now, we are in the teleological argument for God's existence. This is also known commonly as the argument from design. In the last session, I discussed that the argument from design, at least in my mind, is a series of arguments rather than just one. In the last segment, we talked about the lack of evidence for abiogenesis, which is the origin of life from non-life. Today, I'm going to talk to you about the next sticking point that works against atheism, and that is the presence of DNA. And of course, as a physician, and especially one that deals with cancer, I am very familiar with DNA, the information that it carries, and the implications of errors in its formation and replication. So let's talk about the three main things that point to the personal creator God in relation to DNA. The first thing in DNA that points to God as the creator is the origin of the molecule itself. So let's look at the structure of DNA. DNA is a very complex molecule whose structure was elucidated in 1953 by Nobel laureates Watson and Crick. DNA is made up of building blocks called nucleotides. There are three components of a single nucleotide: a deoxyribose sugar, one phosphate, and one of the four bases. To make a complete DNA molecule, single nucleotides join to make chains that come together as matched pairs and form long double strands. DNA normally exists as a double-stranded molecule. In living things, new DNA strands are put together always using a pre-existing strand as a pattern. A complete DNA molecule has two side-by-side polynucleotide strands twisted together. Bases attached in the pairs in the center of the molecule, sugars and phosphates on the outside, forming a backbone. When inside a cell, the two strands of DNA gently twist around each other like a spiral staircase. The anti-parallel arrangement of the two strands is what causes this twist. Because the strands run in opposite directions, they pull the sides of the molecule in opposite directions, causing the whole thing to twist around itself. Most naturally occurring DNA strands spiral clockwise. A full twist or complete turn occurs about every 10 base pairs or so, with the bases safely protected in the middle of the helix. Perhaps the most important thing is that DNA uses a pre-existing DNA strand as a pattern or template in the assembly process. DNA doesn't just form on its own. The process of making a new strand of DNA using a pre-existing strand is called replication. In the last video, I talked about the near mathematical impossibility of the formation of life-containing and life-defining molecules out of inanimate and random molecular interactions. Second, let's talk about protein synthesis and replication of DNA. Now you may say, wait, if we have DNA, we have life. First of all, that's simply not true. Although DNA contains the information for the inner workings of the cell, DNA by itself is not life. In fact, in the last video, I talked about one of, if not the simplest life forms, the mycoplasma bacteria. Even that simple single-cell organism contains three-layer cell membrane, soluble proteins in the cytoplasm, ribosomes, DNA, and RNA. Furthermore, even if somehow a cell came together with DNA in it, but no way to replicate, so that the cell would divide and then the DNA is just useless. So what does it take for DNA and the cell to carry out protein synthesis and to replicate? Well, obviously the DNA molecule is needed, but so are ribosomes and RNA. So here are the bare bones minimum of protein synthesis. The message contained within the mRNA is converted to protein through a process called translation, where the genetic code is deciphered into amino acids. There are many important players in protein synthesis, but the two in particular have crucial jobs. Ribosome is a molecule whose job it is to hold everything in place as well as form the bonds between the amino acids. All cells have ribosomes, and they are made up of RNA and associated proteins with a small subunit and a large subunit coming together during translation to catalyze or speed protein synthesis. Transfer RNA are small RNA molecules that are folded into a specific shape necessary for fitting into ribosomes, carrying an amino acid and reading a codon. The way that each tRNA recognizes a codon is through base pairing with a complementary sequence on the transfer RNA called the anticodon. Initiation or beginning of protein synthesis involves assembly of the ribosome, the tRNA that recognizes the start codon, and the mRNA molecule itself. A second tRNA for the next codon enters the ribosome, and the first two amino acids are joined with a peptide bond. Elongation happens as the ribosomes moves along the mRNA so that tRNAs can enter, add the appropriate amino acid to the growing peptide chain. Termination then occurs once the ribosome has reached the stop codon. At this point, the ribosome separates into its two subunits, and the mRNA molecule and the peptide chain are released. So how does this relate to the topic at hand? Simple. Ribosomes themselves are minute particles consisting of RNA and associated proteins found in large numbers in the cytoplasm of living cells. They bind messenger RNA and transfer and synthesize polypeptides and proteins. But they must exist in order for protein synthesis to work. So where did they come from? The problem is DNA codes for proteins and RNA contained in the ribosome. So how's this possible when the two are reliant upon each other? You see, this makes the system one of irreducible complexity. In other words, all the parts in their finished form must be present in order for the system to have any function at all. Thus, the system cannot be accounted for using abiogenesis nor neo-Darwinian evolution. DNA replication is even more complicated. The steps involved in DNA replication must happen in a precise order. 1. Supercoiled double-stranded DNA is relaxed by a special enzyme called topoisomerase or gyrase. 2. The DNA is then unwound by an enzyme called Helicase, which opens up the two strands in one area at a time. 3. Nucleotides matching the bases exposed by the unwinding base pair with their match. 4. The enzyme DNA polymerase then joins these bases together by catalyzing the formation of a bond between the phosphate of one nucleotide and the sugar of an adjacent nucleotide. And if you think this is a simple process, it is not. You see, this enzyme even has a proofreading function, correcting any mistakenly added nucleotides. 5. The enzymes move farther along, unwinding the next section of DNA so that more nucleotides can join the growing chain of the new DNA strand. So you can see, this again is a process of irreducible complexity. The parts in their final finished form must all be present at the same time in the same location in order for the processes to take place. And if it doesn't, then the process fails, the cell does not replicate, and the cell undergoes pre-programmed cell death. Hopefully, at this point, you're starting to see the problems with atheistic materialistic explanation for these processes. Finally, let's talk language. What do all languages have in common? 2 things. First, all languages transmit information. Second, all languages are produced by intelligent design. If you don't believe me, then consider this. You're walking down a city sidewalk and look down to see a flyer advertising a concert at City Hall tonight at 7:30 p.m. You would never in a million years think that the ink had randomly landed on the page, unguided, in a manner that conveyed the kind of specific information on the flyer. Let's take it one step farther. Imagine you're walking down that same street and instead of a flyer, you see a book. You pick up the book and lo and behold, it's Romeo and Juliet. Would you ever think that those words landed on the page in that particular order on their own? Of course not. For goodness sakes, even if you put 1,000 of the most intelligent chimpanzees in the world in a room full of fully functioning computers, they would never complete even a page of Shakespeare, much less the entire thing. So how does this relate to DNA? Simple. DNA transmits specific complex information, as we just discussed. So how much information does DNA contain, you ask? Well, the DNA in a single amoeba contains more information than 1,000 volumes of the Encyclopedia Britannica. And that's just a single cell organism. What about the 23 base pairs of chromosomes of the human genome? Well, they contain all the information that drives cells to become brain cells, some become kidney cells, some red blood cells, and on and on and on. Quite frankly, the amount of information is absolutely incomprehensible. Even the parts of the human genome that were once thought to be nonsense segments are now known to be important for signaling. So in summary, everything about DNA, from its incredibly complex and specific structure to the irreducible complexity of both protein assembly and DNA replication, to the immense amount of information contained within the DNA molecule, all indications point to a purposeful, personal, all-knowing Creator God. I hope this segment has given you some evidence for the existence of God. In the next episode, we'll be discussing more details of the concept of irreducible complexity. If you like the content of today's video, please give us a thumbs up and be sure to subscribe and hit the bell to be alerted to future videos. Thank you and God bless.